Okay, live stream is on. I've gotten so used to giving thumbs up like that. I'm pretty sure now I do it in person. It's like, okay, yes.
I don't think that's weird. Maybe I'm just a big dork, but I definitely give people thumbs up in real life.
But this is something I have acquired during this whole like virtual thing.
Right, because you got to communicate just without having to unmute. Yeah, it makes sense. No, I don't know. I don't think it's weird.
I'm also team thumbs up. I'm also thinking that it makes me look like a crazy nerd, but it's okay.
But that's okay because we're surrounded by them, so no
it's
judgment.
True. Those are our people.
All right, cool. So it looks like, thanks, Natalie. It looks like YouTube is a go. So Ankita, whenever you want to start.
Okay, I think we should start because we are 11:01. Okay, welcome everybody for another session of Cell Migration Seminar. It's always so wonderful to have you all here. We have two awesome speakers today. The first one is going to be Dr. Michelle Baird. She is a postdoc fellow in Dr. Claire Waterman's lab at National Institutes of Health. Also pretty much my neighbor and colleague. I spend a lot of time next to Michelle. She received her early training in a National High Magnetic Field Lab in the lab of Dr. Michael Davidson, focusing on the development and optimization of fluorescent proteins for live cell microscopy. After witnessing the power of light microscopy to understand cellular behavior and function, she went to pursue her PhD in biophysics at the University of Maryland, co-mentored by Dr. Kimberly Stroka and Dr. Claire Waterman. There she focused on understanding how transcriptomic changes associated with melanoma progression influence cell migration through complex 3D environment and tumor genetic heterogeneity. And I think that's what she's going to talk about today. And she's really passionate about scientific outreach and training in career development, but she was talking about her work in ASCB. So the floor is all yours, Michelle, and we are looking forward to your talk.
Well, thank you so much for that very kind introduction, Ankita. And thank you to both you and Jen for allowing me to talk about my PhD work. I'm just so honored and thrilled to talk to everybody today. It's such a great seminar series with such a wonderful and engaged audience. Let me go ahead and share my screen. Okay, good. We can see everything's good?
No, yeah, all good now.
Okay, so thank you again, everybody, for having me, and I'll just get right into the talk. And today I'm going to talk about nuclear fragility in cases of confined migration, particularly in melanoma tumors. So there we go. So I don't need to introduce to this crowd at all the just so fascinating and amazing world of cell migration and how many mechanisms and modes they can use depending upon their state of being. It's just a wonderful thing. And today, however, though, I want to focus on some of the consequences of cell migration. So in these beautiful intervital movies from a few years ago from Peter Friedel's group and John Condelis, we look at cells migrating through these complicated topographies. So all of these examples are melanoma cells in a mouse dermis. And I hope that you can appreciate from either these top two videos where it's single cells migrating along collagen fibers using MMPs and other things like that, or if it's this more collective invasion down here that they're following their tracks with leader cells and other cells, that the cells have to migrate through this really complicated environment. And the pore sizes are anywhere from, a few microns to 30 microns in diameter. So they're going to have to go through places that are really tight squeezes for them. So it wasn't until this landmark paper from 2016 where we really appreciated what the consequences of the cell to go through these confined areas are. So these are both examples of melanoma nuclei migrating through a mouse skin dermis. And in this movie right here on the left, you can see that there is a soluble GFP that's tagged with the nuclear localization signal. And this is in the nucleus dissolubly around. And then when there is a rupture or a tear in the nuclear membrane, it causes the soluble marker to spill out into the cytosol of the cell, which you can see quite dramatically here. And this is a great visual cue to indicate that the nuclear membrane no longer has integrity, and there could possibly be mixing of the nuclear and the cytosolic compartments. So further labeling with lamins and histones shows that these are not just tiny pinpoint ruptures, but instead are these large-scale herniations almost of the histone complex, which is shown here in red. And this is all now exposed to the cytosol of the cell and could lead to potentially catastrophic consequences. So work from several groups such as Dennis Disher and Matthew Piel's lab have really come to terms with a nice, clear step-by-step mechanism for how this roughly occurs. So when you have a cell migrating through a confined area or a tissue, you can get this incidence of a nuclear bleb, and this is primarily pressure driven. And then when this confinement continues and persists over time, this nuclear bleb has a tendency to rupture. And this leads to mixing of that cytosol and nuclear compartment and has been shown to lead to DNA damage. So the primary mechanism, which is thought to occur, is through DNases that are present in your cytosol. And this is something that protects your cells from foreign DNA, such as from viruses or things of that nature. So they're normally there as part of your immune surveillance. However, when this integrity of the nucleus becomes compromised, they can come in and they can attack your own DNA as well, not recognizing that it's itself. So over time, the membrane eventually becomes repaired through the escort complex. And this is the exact same mechanism that's normally used during mitosis to reseal the nuclear membrane when the nuclear envelope reforms. However, that DNA damage remains in the nucleus. And over time, if this repeatedly happens, that DNA will persist. And if it's improperly repaired through issues with repair pathways, as is known in many cancers, it becomes heritable from the next and next generation. So this has primarily been looked at with the players that I previously mentioned, and then also the interplay from the nuclear cytoskeleton, so such as the lamins. The lamins are known to have a really strong influence on nuclear envelope integrity and mechanics, and a lot of the early research has been done with a particular focus on them. However, I hope you can appreciate from this cryo-EM that the nuclear envelope is a mass of complicated proteins, many of which are totally uncharacterized. So with this in mind, we really got to our first two big questions in part one of my talk, which is if we could identify if nuclear envelope genes are transcriptionally altered in patients during cancer progression, and if this could somehow play a role in modulating that nuclear fragility that we're seeing and genetic heterogeneity in that tumor cell population. So we wanted to get at this completely blindly. So we chose melanoma as a really nice model system, and we liked it because it's biopsied clinically quite often. So we have a really good transcriptomic database of all of these patient tumors from really clear clinical stages of the disease progression. So we used two published RNA-seq databases of melanoma tumors from early stages, so stage 1 benign nevi, as well as much later stages, such as stage 4 and already metastatic to the lymph node data sets of either full complete melanoma tumors or also FACS-sorted isolated melanoma tumors. And we use this as well as a combination of advanced melanoma cell lines from Berg CCLE, as well as our own sort of model cell lines of choice, 1205 Lus, to really try to get a picture of the transcriptomic changes that are occurring as the disease progresses from early stages to much, much later stages. So with that all in mind, we had around 3,000 gene candidates that were all consistently altered in the more advanced melanoma compared to the benign stages. And we use gene ontology databases to fragment those into just the nuclear genes of interest. So things involved in histones and nuclear envelope and nuclear membrane and in the ER nuclear envelope connections to try to blindly get at possible candidates that would alter nuclear envelope fragility in cases of this confinement. So with that list, we found around 10 genes that we all were really excited about as potential possibilities. And when we went back to our patient tumor data set, we found that they all really nicely became upregulated when you compared the earlier stages to the later stages of metastatic disease. So all of these genes are increasing as the cancer progresses. So with that, we decided, since they're all upregulated, that we are going to do an siRNA-based screen. And with that, we can reduce those already high levels of genes down to where they would more normally be in a benign cell. So we also wanted to approach this in some sort of standardized assay that we could reproducibly confine the cells in order to explore whether any of these proteins had an effect on nuclear envelope integrity when the cells are confined. So to do that, we used a PDMS confinement device, which will reproducibly confine the cells to 3 microns over time, and then allow us to observe what happens to the cell as a consequence of confinement. So if we first look at our benign cells, our benign melanocytes, let me go back. We see using our 2 markers, the NLS marker that I described previously. Oh no. Okay, there we go. We see that in the incidence of confinement that there's no NLS leakage, the envelope appears intact. Whereas when we looked at our malignant melanoma cell lines, we see catastrophic rupture as a consequence of confinement. We see leakage of that NLS signal, which is magenta here, but we also see localization of this other sensor, a DNA sensor called cGAS. So this is part of your STING pathway, which again also is in the cytosol of your cell and provides immune surveillance to foreign DNA that would enter your cell. But in this case, we're using it as a really nice and clear sort of biosensor indicating that the DNA has been exposed to the cytosol of the cell. So these are not just small ruptures. Again, we're having that DNA exposure, which has been linked to the DNA damage. So we performed our screen of our sort of high priority targets. And to our surprise, there were 2 that when we reduced their high levels in our cancer cells, we saw a dramatic decrease in the amount of rupture frequency that we observed. So in particular, Lamin B2 and Lamin B receptor dramatically decreased the number of cGAS foci that we saw as an incidence of confinement. So somehow these high levels of Lamin B2 and LBR in our cancer cells appear to be modulating the fragility as the cells are confined. So we decided to, in particular, focus on Lamin B receptor. And so just to follow up a little bit about what Lamin B receptor is, for those of you who don't know, it's a super fascinating protein. I'm completely obsessed with it. It's very cool. So it resides on the inner nuclear membrane of your nuclear envelope, and it has kind of 2 main functional jobs in the cell. So its N-terminus directly and indirectly binds to lamins, such as Lamin B, heterochromatin modifications, as well as histones H3 and H4. And it has 2 sort of really cool processes that it's involved in. So first off, when the cells are dividing and then the envelope reforms, LBR has a really vital role for helping to establish that peripheral heterochromatin that's needed to reform the nuclear envelope. It also has a really interesting role in development, where a lot of the cells, once they become differentiated, are normally high in LBR, and then that switches sort of off with Lamin A as the cells become more differentiated over time, and they trade off roles in tethering that heterochromatin to the nuclear envelope periphery. So I found that really fascinating. And then the second, even more interesting part of LBR is at its C-terminus. It has this C14 sterile reductase domain. So for those of you who are not super familiar with cholesterol synthesis, because I was not, This is part of the component that's required to synthesize cholesterol in your cells, part of the synthesis pathway, and it is required to generate cholesterol at the end. So you normally have two in your cells. You have one that resides in the ER, and then you also have the Lamin B receptor, which has this interesting location in the inner nuclear membrane. It also is associated with several human disease conditions. If you're heterozygous for a series of point mutations, it leads to these neutrophil defects that are primarily characterized by nuclear shape, where the nuclear shape becomes more abnormal when you have these defects. And then also, if you're homozygous for these defects, unfortunately, that is embryonically lethal and results in a series of metabolism-related defects in utero. So with that in mind, I want to go to sort of the second half of the talk, where we're really interested in trying to figure out how LBR is particularly playing a role in nuclear envelope fragility. So we first wanted to ask if LBR itself is sufficient to induce nuclear fragility in high quantities, if it's the result of alterations in nuclear mechanics, which is similar to what's been published previously with things like Lamin A. And then also, what is the specific molecular mechanism of LBR's role in this nuclear fragility response? So to get at our first question, we took our benign melanocytes and we just overexpressed LBR. And first, I want you to just compare looking at just ER and CGAS. Again, like I showed before, we don't see any rupture in our benign cells. However, when we overexpress LBR in this movie below, you really see that the rupture is catastrophic. And hopefully you can appreciate it's even somewhat different than the 1205 Lu rupture in the melanoma cells, where we're not seeing those really nice distinct foci events. Instead, we're seeing what looks almost like a perpetuating cracking around the nuclear membrane, where there's lots of sites where the C-gas is now able to access the DNA. So we found this really fascinating, and I'm just going to show the quantification here, where again, just adding extra LBR is sufficient to make them much more fragile, and basically to the same degree that we see our cancer cells when they're exposed to confinement. So we thought this was super cool. And our first hypothesis is this is totally something to do with nuclear mechanics. Somehow LBR is changing the susceptibility of the cells to either withstand or tolerate this level of confinement. So to get at this, we decided to use atomic force microscopy-based assay, or AFM. So this is a cantilever tip that has a 25 micron bead attached to it. And we use live cell microscopy to basically align the cantilever on top of the nucleus of adherent cells. And then we do a brief deformation and we measure the mechanics that we see. So I'm going to show an example force curve right here. And when we indent the cell, I hope you can appreciate there's sort of two different force regimes that we see. We see an initial force regime, that is the plasma membrane or the cortex element. And then as we continue to deform the cell, we eventually reach the mechanical component of the nucleus, which has a much more linear sort of curve to it. So when we do a line fitting to just this linear regime, we can extrapolate the effective nuclear stiffness of these cells. And our first observation was the melanocytes actually have quite a stiff nucleus, somewhere around like 58 piconewtons. However, that's almost cut in half when we put in extra LBR. So LBR appears sufficient to soften the nucleus and make it more deformable. So similarly, when we look at our cancer cells, we see that the cancer cells have quite a soft nucleus, which really correlates nicely with the fact that they have this high LBR expression. And indeed, when we knock down that LBR expression, we see this stiffening increase, again, almost to the same level as our benign cells. So it really appears that LBR in particular can rapidly modulate the softness and the stiffness of the nuclear mechanics, as well as affect the fragility of it in terms of confinement. So we now wanted to get into, what is the molecular mechanism of this entire thing? So I just want to remind you briefly of all of our functional domains that we have. So there's the N-terminal domain and the C-terminal domain. So we decided to use a series of protein truncations and point mutations to particularly perturb each one of these segments. So if we first focus on the N-terminal domain, we cut off the tutor in the RS domain, and we found that it localized correctly, but it could no longer bind that chromatin in the lamin. And this we thought for sure was going to be responsible for the change in nuclear mechanics and fragility that we're seeing. Based on all the literature, we thought 100% it was something to do with the chromatin and the lamin binding. However, just to make sure, we also used a series of point mutations that are found in the patients of the diseases that I described previously, and have been shown to completely obliterate the cholesterol synthase activity of LBR. So just to check, we decided to use those as well and see whether it was anything to do with the C-terminal domain. So we used our two assays. We used our confiner assay and our AFM, and to our complete and utter shock, the N-terminal domain truncation did not really appear to have a role in our nuclear envelope fragility. When we rescued with it, it was basically the same as the wild type 1205 blue, indicating that that N-terminal chromatin and lamin binding was not required for the change in fragility that we observed. However, when we used both of our point mutations, we found that this did not rescue the fragility response, that we were seeing incredibly low nuclear envelope fragility similar to our LBR knockdown phenotype, indicating there's something particular about the actual activity of LBR in being able to synthesize cholesterol that leads to this nuclear membrane fragility. So similarly, when we looked at our AFM, we found that the point mutation for the cholesterol had a similar softening effect. So we really think there's something specific about the ability of LBR to synthesize cholesterol that leads to this change in nuclear mechanics and nuclear fragility. So I'm now going to get into a little bit of preliminary data and explain kind of how we're trying to think about this. And I'm open to questions or thoughts or feedback on any of this because it's all new to us. We pulled up some data that was one of our first observations when we first did the LBR knockdown screen. And we found that compared to our wild type cells, when they experienced a rupture event, we saw a small rupture in the inner nuclear membrane. So here I'm using LAP2 to mark the inner nuclear membrane. However, there was still NLS leakage into the ER, which was contained by the outer nuclear membrane, which once that popped was when we finally saw a rupture event. So I'm gonna slow this down a little bit so you can hopefully see. So this is the inner nuclear membrane, and we're seeing these discrete rupture hole events in the inner nuclear membrane that lead to the spillage of this NLS signal, which is then contained still by the outer nuclear membrane. And then once that ruptures, that's when we see the C-gas accumulation and the NLS leakage. And this is completely different sort of stepwise series of events than what we observe with the LBR knockdown. So in that case, we're seeing much, much smaller sort of discrete rupture little blebs compared to our wild type. And they are positive for both the inner and the outer nuclear membrane. And then once this ruptures, that's when we get the small percentage of cells that do rupture. So this really led us to believe, okay, maybe there's something specific about the inner nuclear membrane that's leading to this change in rupture dynamics that we see. So given the importance of cholesterol in both of our assays with LBR, we decided to try to image cholesterol in the nuclear envelope, which is not easy, let me tell you. So this is a BODIPI labeled cholesterol, and these are confined cells, confined to 3 microns. And I hope you can appreciate that in areas of blebbing and rupture, there's this sort of local accumulation of cholesterol at the neck of the bleb. And that's not something that we see in the LBR knockdown cells. It appears a lot more homogeneous, and we don't see this concentration at sites of rupture. So this is something that we're actively quantifying now and trying to get some more evidence of, as well as whether the total cholesterol levels in the cell are changing due to LBR presence or absence. So just to sort of summarize how we're thinking about this, we think in the case of melanoma, where there is a high amount of LBR, we're seeing sort of homogeneities in the nuclear membrane and maybe points of fragility or weakness due to this localized accumulation of cholesterol. And when the cells are confined, that local area of weakness or fragility can lead to this sort of blebbing response, where there's a bleb that forms outside of the nuclear envelope. And over time, it expands due to the inner nuclear membrane rupturing into the outer nuclear membrane, until that eventually ruptures as well, leading to the flooding of the DNases that cause the DNA damage. And while this is eventually repaired through the S4 complex, that DNA damage remains. And we think that this is really another mechanism for generating that genetic heterogeneity that melanoma is so well known for, and also is what makes it so hard to clinically treat once it gets to the stage. So with that, I really like to thank the Waterman Lab for so much thoughts and feedback and opinions and advice, as well as all of our collaborators, and for you guys for your attention. Thank you so much.
Thank you so much, Michelle, for such a wonderful talk. We'll give people a few minutes to trickle in the questions. Jen, you have a question?
Yeah, I do. So this is probably very naive, but I guess I'd assumed the physical picture that I had in my head was that nuclear resistance to deformation should have a lot to do with the fact that it's packed full of really dense chromatin, right? Like there's just a whole lot of stuff in there, and that should be mechanically resistant. And then your story is focusing, as what's happening right at the membrane. And so I'm wondering if you can help me understand if my physical picture of, how important that, the sac full of stiff stuff is versus the material of the sac itself. And am I just, is that just another part of the story that's adding to it? Or, if you can help me sort of physically understand what's happening here.
So you're 100% right. So Denny Wirtz and Andrew Stevens have really been the landmark labs that have looked at the role of chromatin, and definitely having that densely packed heterochromatin, especially at the periphery, changes the mechanics of the nucleus. And they also are changing it in different ways. So if you think of the lamins as providing more sort of elasticity, whereas the chromatin is providing more of like an initial resistance, and then it breaks. So those are kind of the two ways of thinking about how those components work together to facilitate, deformability or rupture. So I think in terms of the membrane, that's something that no one's ever, to my knowledge, really explored as a component of this entire cycle of things that are protecting the DNA. And I don't think that it's necessarily performing the same degree of resistance as the intermediate filaments or the chromatin is, but I do think it's an unknown right now, sort of what sites generate blebs. Can we predict where a bleb or a rupture site would occur? And I'm wondering if maybe looking at the membrane is a way that we can think about, sites of weakness that would be releases for the pressure that's building up due to this confinement.
Right. And these cancer cells are just in a state where maybe they have disrupted or abnormal amounts of LBR. And so they're not going to be able to respond normally. Yeah, fascinating. Thank you so much for that.
Yeah, of course. Thank you for the question.
Thank you, Michelle. We have next question from Ernest Liu, and Ryan will get to your question after that. So question is, does LBR regulate the nuclear pressure?
Not to my knowledge. There's no evidence of there being a mechanism for it to do that. However, I think there's a lot of things we don't know about how pressure is regulated and changed in the envelope, especially as a consequence of confinement. So, there's always a possibility where somehow LBR could be disconnecting itself from the lamins and the chromatin or something like that would allow for more membrane to be more flexible. So I don't know of an explanation to address that question, but I think that there's always a possibility.
Wonderful. Ryan, if you would like to ask a question.
Yeah, I'd love to. First of all, love the presentation. So great. And I just love how you got the setup to visualize these changes happening live. That's so powerful and really interesting to see. Maybe a question that's going to call for some speculation. I'm really, keen to understand how cells decide how to move. And one of those mechanisms kind of appears to hinge on mechanotransduction of the nuclear envelope. I'm just wondering, any speculations on how the mechanisms you're describing maybe could impact those kinds of choices in how to migrate? Like if maybe I'm thinking predominantly of that wrinkling of the unwrinkling of the nuclear envelope causing the link to the contractility from the Peel lab, but there may be others. So just any speculations on that maybe melanoma would respond differently to a physical environment than other cell types?
So I quickly answer, I totally think yes. And then my speculation on that is based on neutrophils. So neutrophils have very, very low Lamin A and very, very high LBR. And that's what's thought to lead to this really bifurcated nucleus that's really squashy and can easily migrate through confined areas. And the mutations that occur that decrease LBR in neutrophils make this sort of more cylindrical, hypothesized more rigid, but never actually been proven nucleus. So I think all these things are in interplay. So all the Lamin isoforms, LBR, all of that is in various ratios in various cells. However, in neutrophils, if they only have LBR, LBR itself can change that nuclear structure and ability to migrate through confined areas. So I think in melanoma cells, and mine in particular, due to having high amounts of LBR, it screws up that ratio. And maybe if there also was an issue with Lamin A due to, heterogeneity in the population or whatever, we'd see that effect more strongly. So I think in general, yes, that this could be a way to help mechanically transduce that force from external inside of the nucleus through its connections to various nuclear envelope components.
All right, thank you.
Mm-hmm.
I have and again, this is probably very naive, but sort of a follow-up, I think, to Ryan's question and your answer. So is there evidence of mutations in other components that would affect how much cholesterol you have in these membranes that would, I don't know, are those mutated in other cancers or in melanomas? Like, are there sort of unrelated, not necessarily having to do with lamins and sort of the structural proteins making up the, in the membrane, but the cholesterol itself and having to do with the stiffening there?
So
I don't know if that made sense.
For nuclear cholesterol or cholesterol in general?
I guess nuclear. I mean, in general, maybe, but yeah.
So in general, there's a ton of issues with cholesterol synthesis in cancer. And there's been a lot of anecdotal evidence that the use of statins, which decrease cholesterol overall, help you better fight cancer. So people have longer longevities, less metastasis. So there's some anecdotal evidence that cholesterol in general can play a role in making cancer better or worse. And more aggressive cancers may have more cholesterol due to a variety of factors. The cholesterol in the nucleus is sort of a new field. It's very, very hard to isolate any lipid components from the nuclear envelope because the pool there is quite small compared to the plasma membrane and every other organelle in the cell. So there's not a lot known about sort of local concentrations or local sequestering or how this could change as a result of disease state. And there's also no other known component that synthesizes cholesterol in the nuclear envelope. The one that you normally use to all your cholesterol is in the ER. So it's kind of unclear whether the local nuclear cholesterol has any, bigger picture role or has a role in even like the sort of membrane fragility response. It's still really early days for me to speculate. And if anyone has any better ideas on imaging cholesterol, I would love to hear them because it has been challenging.
Thank you, Michelle. I think we will take the question from Alexis, and there's another question for you in the chat. You could probably respond to that because we have to move on after that. Alexis, please go ahead.
All right, very beautiful presentation and sea gas movies. Jesus Christ. Very nice. I was just about to write in the chat to everyone, there's this really crazy paper from the 70s where doctors were trying to install paper filters with different pore sizes onto patients suffering from rare genetic disorders that actually linked to LBR gene. And what they found is that neutrophils that have this sort of really abnormal nuclear shape, they have this really enlarged, rounded, sort of rigid nucleus, they have a lot of problems to accumulate in the paper filter that has specifically very small size of the pores, of the openings. And I found it really interesting, really intriguing. And back in the 70s, it was possible to do experiments in human patients. Of course, these days we can't, but I found this really historical reference to this remarkable experiments. And that's why I wanted to share it with everyone.
No, that's fantastic. Thank you so much.
It's very cool. I think your functional sort of idea, how you can translate all this changes in nuclear mechanics, is that you somehow increase probability of nuclear rupture and therefore nuclear damage. And you will accumulate a lot of nuclear damage and potentially trigger so-called chromatopsis, something that David Pellman is currently pushing forward quite aggressively. And I was wondering, do you think it's possible for you to go back to your first slides where you were doing some bioinformatics magic and see whether you can identify signatures of chromatopsis in melanoma cells, and whether those signatures are somehow associated with changes in LBR gene expression or mutations, and this can provide you with a more solid foundation to speculate about this functional role of increased DNA damage, increased mechanical instability of the nucleus downstream of this LBR pathway. Do you think you can, because it's a relatively cheap, quick, and easy, way to guide your experimental intuition where to probe further?
No, that's a fantastic comment. And I think that I can definitely look back in all of those bioinformatics data sets and see if we can correlate, any specific markers to LBR increase or decrease. And something that's super interesting to me is, there's like some preliminary evidence, if you look at survival curves, that high LBR tends to correlate with low survival in the early stages of tumor genesis. So if they're looking specifically at like stage 1, stage 2, stage 3, getting into metastatic, and then it seems to be less important after it's metastasized and the survival curve doesn't change. So I think that maybe also helps with the thought process that maybe LBR is a double-edged sword, that, it may be great for that initial tumorigenesis response, but you're not going to want to keep it around after the cells form a secondary tumor.
Right. And that's why I think what I was missing in your talk is some functional readouts. For example, what would be the cell fate in the situation of mechanical stress? Will the cell die, survive? How can we modify the cell fate decision? I think it would add more sort of functionality to the work. We can talk later.
We should talk later. Wonderful thoughts. Thank you so much.
I'm in love with your work. Good work.
Yes. Thank you.
All right. Thanks for that, Love Fest. We're going to move on now. Thank you, Michelle, for that fantastic talk. Meta, go ahead and share your screen. And so we are delighted to have our second speaker today. We have Dr. Medha Pathak, who is an assistant professor in the Department of Physiology and Biophysics at the UC Irvine School of Medicine. She obtained her doctoral degree from the University of California at Berkeley and did postdoctoral work at the Harvard Medical School and UCI. And so her lab uses an interdisciplinary approach to understand how mechanical forces shape cellular processes, combining techniques from ion channel biophysics, cell biology, stem cell biology, and bioengineering. She is the recipient of the Helen Hay Whitney Postdoctoral Fellowship and the NIH Director's New Innovator Award and an HHMI Gilliam Mentor. So leave some awards for the rest of us, Medha, and please go ahead. The floor is yours. We're really excited to hear about your work today. Thanks.
Thank you so much, Jennifer and Ankita. It's really an honor and a pleasure to share our work in this seminar series, which I've really enjoyed over the last 2 years. So I'm fascinated by how cells are constantly interacting with their environment and how, based on the information that they receive, they make decisions such as where and how to migrate. So the mechanical cues that cells encounter in their environment are very diverse. They can range from shear stress from the fluid flow surrounding them to strain forces as cells attach to each other and pull and push in each other. And in addition to these dynamic forces, they can also be biophysical properties of the substrate, such as the stiffness of the extracellular matrix and also the nanotopology or the texture of the matrix. And all of these mechanical cues have been shown to powerfully impact cell behavior. But for mechanical forces to influence cellular pathways, they have to be converted into electrical and chemical signals. And this is done by specialized proteins known as mechanotransducers that convert the mechanical forces or signals into electrical and chemical signals. So one class of mechanotransducers is mechanically activated ion channels. And my lab focuses on these because they are amazing first responders, signal integrators, and information encoders. So they have the ability to respond really fast in sub-millisecond timescales to molecular scale forces. And they have a high dynamic range, or rather they can have a large dynamic range, which means that they have the ability to respond to really small signals as well as to really large signals. Their activity generates both electrical and chemical signals, and features such as inactivation or adaptation of the channel provides different mechanisms to encode repetitive mechanical stimuli. Now, the activity or the presence of mechanically activated ion channels was first noted in the late 70s in inner ear hair cells by David Corey and Jim Hudspeth. And then soon after, they were also found in chick skeletal muscle, in bacterial cells, and in plant cells. In bacteria and plants, the channels were to identify the molecules underlying the mechanically evoked currents were identified in a few years. But for the vertebrate system, it took a really long time. It was only in 2010 that the first bona fide mechanically activated ion channel family, the piezo channels, was identified by Adam Paraputian's lab. These channels were found to have a remarkable propeller-shaped structure with large blade-like regions that are thought to detect the mechanical signals. The ion conduction happens through a central pore pathway. Let me get my pointer here. So this is the central pore domain, which is through which the ions pass through. And the importance and impact of this discovery, the identification of the piezo channels, was recognized through a Nobel Award to Adam Paraputian this last year. In the last decade, Piezo1 has emerged as a powerful mechanotransducer that orchestrates a number of different biological processes across the human body, as is depicted in this cartoon from your Grandel's lab. The knockout is embryonic lethal, and mutations in the channel have also been found to be linked to a number of human diseases. So my lab studies Piezo1 for the molecular to the systems level, and we started off first asking how Piezo1 is activated by mechanical forces. So the standard approach to studying Piezo1 activity has been patch clamp electrophysiology. And this, of course, is a technique which measures the electrical signals produced by the activation of the ion channels. And then combined with patch clamp electrophysiology, other techniques have been developed to impart a mechanical stimulus to the cells as the electrical measurements are being made. And a number of studies doing this in different ways have demonstrated that Piezo1 activates in response to a very large diversity of mechanical cues. In fact, almost anything mechanical that you can do to a cell is likely to activate this channel. But cells also generate mechanical forces. And just like you or I would poke or pull on an object to determine whether it's soft or stiff, cells do something similar. They attach to the substrate through focal adhesions, and then the actomyosin cytoskeleton that is attached on the intracellular side of these generates a mechanical force that tugs on the substrate to determine its stiffness. And traction forces and substrate stiffness determination are important for several biological processes, including stem cell fate, cell migration, wound healing, as well as cancer progression. But the molecular mechanisms by which traction forces influence these processes is not very well understood. So we wondered whether traction forces could activate the Piezo1 channel and whether its activity could then regulate downstream signaling processes. So to study this, we developed a new approach to measure Piezo1 activity in intact cells without disturbing their architecture or their cytoskeletal dynamics. So we took advantage of the fact that the channel lets in calcium, and so we can use fluorescent calcium indicators to measure its activity. And then by using TIRF microscopy, or total internal reflection fluorescence microscopy, we can image at the cell substrate interface, which is where stiffness sensing is taking place. So we did a very simple experiment where we loaded cells with a calcium indicator, such as fluorophore or Cal 520, and then we imaged them in TIRF microscopy. This is what we saw. So in wild type cells, we saw a lot of calcium dynamics, but in knockout cells, this was drastically reduced, indicating that we were indeed measuring Piezo1 activity. So and for every region where you see this flash of light or every Piezo1 active site, you can plot a fluorescence intensity over time, which shows these spontaneous calcium transients, which you can then quantify in terms of their amplitude and frequency and so on. So now in this experiment, we are not applying any external mechanical force to the cell. That is, we are not poking or pushing or stretching or squirting fluid on the cell. And yet the channel is active. So we were curious as to where the force is coming from. And we hypothesized that cell-generated traction forces may be activating the channel. Now, traction forces, as we know, varies with substrate stiffness, with harder substrates eliciting larger traction forces and softer substrates eliciting smaller traction forces. So we asked whether Piezo1 activity is also higher on stiffer substrates. So we did our TIRF calcium flicker measurement on cells that were seeded onto substrates of different stiffness. And what we found was that on soft stiffness, there was little to no activity of Piezo1. And then as we increased substrate stiffness, we saw more and more activity. And when we quantified the amplitude and frequency of events, we saw that there was distinct increase with the maximal activity in terms of both amplitude and frequency happening on glass, the hardest substrate that we tested. So now we asked whether inhibition of traction forces affects Piezo1 activity. So we used blebistatin, which inhibits the ATPase activity of Myosin-II, and we found that treatment with blebistatin reversibly inhibits the Piezo1 channel calcium flickers. And so together, these findings demonstrated that cellular traction forces activate Piezo1. So now we were curious as to whether this is global or local. That is the channel activation localized to regions of traction forces in the cell, or is the mechanical stimulus propagated over long distances to elicit a global activation of channels? To answer this question, we collaborated with Ian Parker's lab to implement a super resolution analysis algorithm that his student Kyle Ellefson had developed. And this technique allowed us to make cellular maps of Piezo1 activity. So we can again start with a Piezo1 calcium flicker movie, such as this one, and we can put it through Kyle's flicker algorithm. And very briefly, what it returns to us is a map of active Piezo1 channels, where each active site that we saw in the movie is shown by a white dot. So based on this analysis, it was apparent that Piezo1 didn't seem to be active all over the cell. Instead, it displayed hotspots of activity. So we asked whether these hotspots correspond to regions of traction forces. We teamed up with Alex Dunn's lab to use their FRED-based molecular tension sensor. And again, very briefly, one can deposit a carpet of sensors on cover slips, seed cells onto them, and measure the FRED signal from the sensor, which encodes the traction forces produced by the cell. So we combined our approach to make maps of Piezo1 activity with Alex's approach to make traction force maps from the same cell. And now here, blue depicts regions of high traction forces, And then when we overlaid the piezo1 activity map onto the traction force map, we saw a beautiful correlation between regions of the forces that had high traction, regions of the cell that had high traction forces, and where piezo1 activity was present. So now there are two possibilities for how this happens. One is that piezo1, the protein, is present at focal adhesions. The other possibility is that piezo1 is distributed all over the cell, but only activated at the force-bearing adhesions. So to distinguish between these two possibilities, we harvested cells from a knock-in Mariposa mouse made by Adam Paraputian's lab, where the endogenous piezo1 protein is tagged with a TD tomato reporter protein. And we immunostained both for the TD tomato and for focal adhesions. And we found that the location of the piezo1 protein is not restricted to focal adhesions. So we are dealing with possibility number 2, where the channel is distributed all over the cell surface, but it's only locally activated near the traction forces. So in this work, we learned that piezo1 transduces not just external outside-in forces, as was very well established in the field, but also forces produced by the cell, which are orders of magnitude smaller. And this occurs in a spatially regulated fashion, which indicates that cells have a built-in mechanism to spatially segregate mechanotransduction events. So having found this new modality of piezo1 activation, we were, of course, very interested in understanding the physiological impacts it may have. And in the last few years, we've found that it's important for stem cell fate, macrophage function, as well as in cell migration and wound healing, which is what I'm excited to tell you about today. And this is work done in a close collaboration with Adam Paraputian's lab. So a hallmark of skin wound healing involves cell migration such that the keratinocytes located at the wound edge migrate into the wound bed to reestablish the epithelial barrier. And this process is known to be mechanically regulated, but the underlying mechanotransducers are poorly understood. So Adam's lab found that keratinocytes express piezo1 and wondered whether it may play a role in wound healing. So in my lab, Jesse Holt, graduate student, first asked whether keratinocytes exhibit piezo1 calcium flickers like we had seen in other aberrant cells. So we did the turf calcium measurements exactly as I described before, and we found that keratinocytes did indeed display the calcium flickers. And as expected, these are reduced in keratinocytes harvested from knockout mice that lack piezo1 in the skin. Adam's lab also made gain-of-function mice. So these are mice that harbor a gain-of-function mutation in the piezo1 channel in the keratinocytes. And when we imaged these cells, we found that they had more calcium flickers, indicating that the piezo1 channel does indeed show the gain-of-function effect in the calcium flickers. Now, to determine whether piezo1 affects wound healing, Adam's lab did a wound healing assay an in vivo wound healing assay on mice, where they made wounds on the dorsal skin and then examined the wound size 6 days after wounding. So in the knockout mouse, they found that at 6 days, the wound sizes were smaller, which means that the wounds were healing faster. Whereas in the gain-of-function mice, they found that the wound sizes at 6 days were bigger, which means that the wounds were healing slower. So in my lab, we asked how manipulating piezo1 may give rise to this phenotype. So we did single cell migration assays where we harvested keratinocytes and watched how they moved. And here is a movie showing control cells on the left and knockout cell on the right. And what we saw was that the knockout cells migrated faster. And this was also apparent in the mean square displacement plots. So to understand how piezo1 may regulate cell migration, we asked, where is it located? We again used the piezo TD tomato reporter mouse from Adam's lab. And when we looked at non-migrating keratinocytes, we found, as before, that the channel was distributed all over the cell surface in a punctate manner. Interestingly, when we looked at actively migrating channels, we found that piezo1 is enriched at the rear. So the rear of the cell is a site of maximal retracting forces, And this observation hinted to us that maybe Piezo1 is involved in retraction. So to test this, we imaged keratinocytes in DIC, and now we focused on the edge dynamics. So we first imaged the cell in a controlled solution, and now we can draw a line spanning the edge of the cell and construct a kymograph, which shows cycles of protrusion and retraction, exactly as we would expect for a migrating cell. And then we added Yoda1. This is a chemical activator of Yoda1, of the Piezo1 channel. And then we continued imaging, and we saw increased dynamics of the edge. When we made the kymograph, we saw that there were more cycles of protrusion and retraction, but there was an increase in retraction, which resulted in the envelope of the cell moving backwards and retracting. And importantly, we don't see this effect of Yoda1 in Piezo1 knockout keratinocytes, indicating that the effect is indeed mediated through the Piezo1 channel. So this experiment is looking at the single cell level, but in wound healing, cells migrate collectively. So how might Piezo1 mediated retraction be playing out there? So we did an in vitro scratch assay where we harvest keratinocytes and plate them in a monolayer, and then we make a scratch to mimic a wound, and then watch as the cells migrate to close the wound gap. So we found that the scratches made in the knockout monolayers closed faster. And a kymograph of the wound edge showed little evidence of retraction. In the knockout monolayers, they processively moved forward. Okay, we did the same experiment with Yoda1 treatment. So I'm showing here control on the left and Yoda1 on the right. And we found that Yoda1 treated monolayers barely migrated into the wound gap. Indeed, in some cases, instead of closing to seal the wound gap, the monolayer actually opened up farther when we activated the channels with Yoda1. And in looking at the kymograph, we see more instances of retraction in the Yoda1 treated monolayer. So to figure out how Piezo1 is modulating collective cell migration, we imaged Piezo1 localization in the scratch assay. So we did 16-hour time-lapse experiments in which we made a scratch and watched it heal. But now we imaged in DIC to look at the location of the cells, and we also imaged in TURF to look at the localization of the Piezo1 channels. And what we saw was that the localization of Piezo1 is dynamic, so it keeps moving, and the channels occasionally form clusters at the wound edge. So there you see a band of Piezo1 clusters forming. I'm going to let this play one more time so you can appreciate how much the distribution of the Piezo1 channel protein changes over time and how it sometimes forms these clusters at the edge of the cell. And there it appears. We stared at these movies long and hard, and then a pattern began to emerge. We saw that in regions where there was no enrichment of the channel at the wound edge continued to migrate forward to close the wound gap. On the other hand, when channel clusters formed at the edge, this was followed by retraction, where the wound edge moved backwards instead of going forward to close the wound gap. So altogether, we learned that enrichment of the channel at the wound edge leads to a local retraction, which then slows wound healing, which is why we see faster wound healing in the absence of Piezo1 and slower wound healing in the gain of function mutants. So Piezo1 regulates the speed of wound healing. And mechanistically, we found that cellular traction forces elicit, we found earlier that cellular traction forces elicit Piezo1 activity. And now we are learning that Piezo1 activity can in turn regulate cytoskeletal dynamics. And thus there seems to be a positive feedback loop between Piezo1 and the cytoskeleton. So more broadly, we demonstrate a role for Piezo1 in cell migration, which is a fundamentally important biological process. And while this is interesting in itself, what really excited me about this work is that it demonstrated that the nanoscale spatiotemporal localization dynamics of the channel can shape tissue scale events. And that's why my lab is so interested in studying Piezo1 from a molecular to a systems level. And we are particularly interested in its role in neural development. a process in which cell migration is really important. And moving forward, we are using human brain organoids to study human development, the neural development, since it faithfully recapitulates several processes that are human-specific and not fully captured in mouse model and animal models. So the organoid system also gives us a great in vitro platform for bridging the molecular and cellular to the tissue scales. And to do this work, we are expanding our team. So if you're excited about interdisciplinary multi-scale approaches to studying mechanotransduction and enjoy working with a dynamic creative team, please reach out. I'd like to end with thanking an incredible group of people, both within my lab and through collaborations that have made this work possible, and also the generous funding sources that have supported our work. So thank you very much for your attention, and I'm happy to take questions now.
Yeah, Ankita, go ahead, please.
I was raising the real hand. Wonderful talk, Medha. I had a question regarding, I always thought piezo needs the stretching, right, in this direction for it to activate. How is traction force, which is in certain angle, able to do that? Maybe I'm not, this might be a real naive question, but I'm not able to conceptualize that. Sorry.
No, that's a great question. It's one that we puzzle over a lot as well. I think the simplest explanation that would be consistent with what's known is that traction forces are somehow producing a localized increase in membrane tension that is activating the piezo channel. But we can't at this stage also rule out the possibility that there is some kind of protein-protein interaction or some kind of a tether-based activation of the channel. I think certainly, going through the membrane tension is sort of the simplest explanation, because we know that piezo1 is gated by membrane tension. But there is piezo1. I mean, this channel is constantly surprising us by all the interesting things that it can do and the many ways that it's regulated in. So it's also possible that there may be more happening than just membrane tension-based activation.
Awesome, thank you.
Mm-hmm.
We have a question from Venkat Maramuthu. Does piezo1 activation increase traction forces locally?
We think it does, and this is something that we are looking at in more detail. So we should have more information on it. Yeah.
There's a question from Nilet Tareja asking, is the frequency or the amplitude of piezo1 channel activity correlated with local focal adhesion dynamics? For instance, sorry, go ahead.
Yeah. Oh, yeah. Again, good question. So we haven't looked at dynamics of focal adhesion in the context of piezo1 activity or expression. There's a recent paper from Robert Mayer's lab where they look at they look at focal adhesion dynamics in the context of piezo1 knockout, and there they find that the focal adhesions formed in the knockout cells, and this was a neural crest cells, they tend to be smaller and shorter lived. So there seems to be more turnover there. And in their study, they also, like us, see faster cell migration in the absence of piezo1.
I think what he's also trying to understand is whether there is some correlation with the channels, let's say, opening more or longer in the regions where there are more focal adhesion.
Yeah. So that's an experiment we're very interested in doing, which is to image so we have imaged piezo1 location in migrating cells, but to also image piezo1 activity. And we tried doing that. It ended up being a technically more challenging experiment, and so we are still working out some of the technical snags. But I think that'll be a very informative experiment. So we're very excited about doing that.
Thank you.
Great. We have a question from Anna Pasapera asking, can you speculate how this transmembrane complex piezo channel is relocalized to the back of the cell?
Again, a great question that we are very interested in figuring out. Don't have answers yet, just a lot of questions.
Awesome. The next question is from Ernest Liu, which was pretty much my question. So I'm skipping that and taking Arminder's Nen's question. Says, very nice talk. Does activation of single channel lead to cascade effect of activation of nearby channels?
Again, another great question. My intuition says yes, but I don't have evidence for that yet. Something that we are interested in figuring out.
Great. It's awesome. I mean, there are just so many open questions here, which is actually really, really exciting. We have a question from Kashish Jain asking, are there any known proteins which might be helping piezo localize to the regions of retraction? So yeah, similar to Anna's question.
Yeah, so we don't know what's regulating the localization of piezo1. What I didn't have time to show in this talk, but we've also found is that the piezo1 puncta are mobile. So even in non-migrating cells on short time scales, if we image the puncta, they are not stationary. They tend to move, they tend to diffuse around in the cell membrane. And again, this was something that was very surprising to me, because if you're designing a mechanotransducer, my sort of simple-minded approach would be to make it stationary, because it's much harder to apply mechanical force to something that keeps moving, right? But the channels are exploring, the puncta are exploring, and we don't know what regulates their mobility. We are, beginning to get little hints on that. And a very interesting question is, what is it that if, as a cell goes from stationary to migrating, what is it that makes the channel protein relocalize in a polarized fashion? Is it just mechanics? Is it just mechanical cues in the cell membrane or the cytoskeleton that are governing where the channels are, how the channels are distributed? Or are there specific protein-protein interactions that are sort of sequestering channels in one domain of the cell? We don't know yet.
Awesome. Next question is from Alexis asking, what is the contribution of wound healing of cell proliferation and versus cell migration downstream of piezo1 activation? And he's citing Goodipati and Rosenblatt from 2017 for that.
Yeah, so Jody Rosenblatt's work showed in a couple of beautiful papers how piezo1 can regulate the epithelial cell numbers and either in terms of cell division if the numbers are low, or extrusion if the numbers are too high. And so we haven't looked at, in our system, in our experiments, we haven't looked at the role of this proliferation per se in the wound healing process. Our experiments are over relatively shorter periods of, the in vitro work is over 16 hours, where there may be some, but it's probably not the dominating factor.
Great. So we have two related questions, so I'll ask them together from Taehyung Kim saying, great talk. Calcium ions also activate myosin activity and increase contractility. So is this what's happening at the rear end of the migrating cells? And then related from Anna Pasapera saying piezo channel activation and accumulation in the back affect myosin accumulation and contractility in the back of the cells. Is that what's happening?
Yeah, so myosin is active when it's in the phosphorylated state. And there are, two major enzyme systems, ROCK and MLCK, that phosphorylate myosin. MLCK is regulated by calcium. So calcium colmorulin will activate MLCK, which will then phosphorylate myosin, make it more active. And so our hypothesis is that piezo1 activity, so traction forces activate piezo1, and then piezo1 activity lets in calcium, which would trigger, which would bind to calcium, to calmodulin, and trigger MLCK and increase phosphorylation of myosin and therefore cellular contractility. So we think that's the feedback loop that's happening. And clustering the piezo channels together serves to sort of harness all of that activity in a small region and generate the localized retraction.
Awesome. Are you okay to answer a few more and stick around?
Yeah, certainly.
Okay, thank you. Next question is from Suvam Mukherjee. Says, looks like piezo is more or less constitutively around in the cell. So he's wondering, since it's not restricted to the focal adhesion, does it just have this mechanobiological significance, or there's something else as well that piezo might be doing?
Mm-hmm. Yeah, so piezo1, we've learned now, transduces, as I just talked about today, the internal forces generated by the cell, but we also know that it transduces a diversity of external mechanical forces that impinge on the cell, right? So looking at it from the perspective of the channel, it doesn't know when mechanical force is going to strike and where, but wherever it strikes, it has to transduce that quickly and efficiently. And so that we think that this distribution that we see of piezo1 everywhere, not just as focal additions, allows it to keep track of both external forces that it has no control over, as well as internal cell-generated forces that are communicated, traction forces that are communicated to the substrate through the focal addition. So it allows it to, transduce both of these efficiently and quickly. And the mobility, the diffusion of the channel that I briefly mentioned, I didn't show you the data today. We think that that provides a mechanism, a biophysical mechanism for the cell to explore, for a small number of cells to explore mechanical microdomains in cells efficiently. So I like to use the analogy of cops patrolling a city for crime, right? A cop doesn't know when crime is going to strike or where, but they need to get there quickly and take care of it. And so there's two ways to do this. One is to station a cop at every block, and then that quickly gets expensive because you have to pay all these cops. Or you can have a fewer number of cops, but give them wheels so they can patrol and cover a larger domain. And so that's sort of my analogy of thinking about piezo1 mobility in cells.
You just resolve crime.
Nice. We have a question. Sorry, we have a question from Carrie and Neeson, asking, does intercellular contact alter local piezo activation dynamics?
A really good question. We haven't imaged piezo1 in the context of cell-cell contacts, cell-cell interaction and additions. But these are regions of mechanical forces as well, where cells are pulling and on each other. So I would totally not be surprised if that's activating piezo1 and piezo1 has functions there. Indeed, there are reports in the literature of it doing interesting things in the context of cell-cell junctions. And that would be something, I think, using the kind of approaches that we've developed of measuring piezo1 localization and activity dynamics in real time as cells and tissues go about their day. I think that would be a really cool experiment to do.
Great. Next question is from Rakesh Kumar Maji asking, does piezo C or the N terminal directly bind to actin or tubulin, or is it some cytoskeletal dynamics during piezo1 mediated just by calcium influx?
There isn't evidence, as far as I know, of piezo1, the protein directly binding to actin or tubulin. And the second part of the question was, so it could be, I think I would say the jury is still out about whether there's some kind of a direct or indirect protein-protein interaction, or whether it's all going through, indirectly, either through membrane tension or calcium regulation.
Great. And you've touched on this a bit, but from Vaishali Mukherjee asking how is calcium signaling from the piezo channels linked to Rho signaling in migrating cells?
Yeah, we haven't looked at this.
Great, awesome. I think we've covered all the questions in the chat. If anyone has a last minute burning question, feel free to pop in. But with that, I'm going to end our live stream. So bye to everybody.